Good afternoon, everybody. Welcome to the latest in our seminar series. Today we have Katerina Poole from Imperial. She is a postdoctoral research associate at the Audio Experience Design team, using methods such as psychoacoustics, electrophysiology, and modelling, investigating how the auditory system processes spatial cues and how we use that knowledge to better render binaural spatial audio. Kat's previous work during a PhD at the UCL Ear Institute uses optogenetics and extracellular electrophysiology to explore how the brain and the single and small networks of neurons in auditory cortex and hippocampus detect acoustic patterns. So without further ado, I will hand you over to Kat, who will no doubt tell you more about all of those big words.
I've only been a postdoc for like a year and a bit, so a lot of this work will be quite preliminary and sort of, more preliminary results and some sort of like methods in progress. And it will kind of have some of my influence from my PhD, trying to bring sort of more neuroscience techniques within the sort of field of acoustics, since I moved from more biology to more engineering work. So I'm currently funded by the SONACOM grant, which is this big consortium that works on trying to revolutionize the way that we do augmented and virtual reality, with our lab sort of focusing on immersive and spatial audio. And specifically, my work is trying to look at objective ways of testing, personalizing binaural spatial audio in the context of HRTFs. Now, to kind of give sort of broad context, and I'm sure you guys have heard this a million times over, but the auditory processing system within our brain is really complex, but it's also very good at being able to separate different sound sources in a busy scene, such as a pub where you've got your mate to the left of you, someone else to the right, the aircon going on in the background, and all this busy noise. And the brain is constantly extracting cues such as frequency, pitch, loudness, and it uses all these cues to better separate the sound sources. One of these cues is the spatial location of a sound source. And that's sort of the cues that I'm going to be focusing on in this talk. So just to give a brief background, there are sort of three main cues that we use to be able to pull out the spatial location of an auditory source. And one of them is the interaural timing difference. So if a sound comes to the right of you, it will arrive at your right ear before your left ear. And this works very well for low frequency sounds because neurons in the brain can phase lock to certain peaks or part of the cycle in the sound wave, and then from there calculate the difference between the two ears. This is not so great for high frequency sounds, so we can use things like interaural level differences, where again, if a sound is coming from the right, it will be louder in the left ear compared to louder in the right ear compared to the left. But again, this doesn't really work for low frequency sounds, which are able to sort of bend around the head a bit more. And this works very well for localizing sounds in the horizontal plane, but for being able to localize sounds that are in the elevation plane and also in front and behind, we can't really use these cues because they'll be the same between the two ears. So that's where our sort of head shape and our weird pinner come into play to provide these monaural spectral cues. So here you can see some examples of some frequency spectra at different elevations for this random silhouettes. And you can see in some places there are specific notches at certain frequencies that the brain will then interpret to be able to figure out where this sound source is coming from in elevation. Now we can describe all this in the form of a head-related transfer function. Here you can see a head-related impulse response for each ear, so left in blue, red in right, and here you can see it's for the location sort of directly to the left of the subject. And we can see that the impulse response for the left ear is larger than the right ear, giving us sort of level difference, and it's also more delayed in the right ear as well. And we can also look at the spectra of this, and we can see how the energy in different frequencies change. And if you look at somewhere like a location that's sort of directly in front of you, but a little bit elevated, the impulse responses look fairly similar here, but when we look at the spectra, we see differences in the frequency. And we can use these HRTFs to render spatial audio binaurally. So if we apply these HRTFs to a specific sound, we can then simulate the location of this sound. And typically people will, or research labs and people in commercial companies will use a generic HRTF. So one that is such like a key markers. Get a model head, it's got mics in, you can record sounds and grab some HRTFs from it. However, this is not everyone's head. Everyone's head is slightly different. Some have smaller sizes. You can see my head here. It's again a very attractive, bold version of me, with the ear shape again being slightly different, and you know, the head size also being very different. And if we look at things like the absolute ITD difference, so here's a little polar plot where you can see the difference between the two ears getting larger as you move further out, and this is for the Kima. And you can see it for mine. There are slight differences, and same for the ILDs as well. So, you know, an ILD change of a certain amount in this direction will not equate to the same place for my HRTF as with the Kima. And you can also look at things like the transfer functions at different elevations. I don't want you to look at this too much, but it's more just sort of looking at the pattern for the Kima. And then we also look for me, and we see again these differences in fine temporal structure. So, you know, there is a reason to try and personalize HRTFs because the morphology of each person really does influence it. And what do these differences actually mean in practice? Well, one big thing is getting localization errors. So you get a lot of front pack confusions because you're not actually capturing the binaural spectral cues accurately when you're using generic HRTFs. This can lead to reduced speech intelligibility because you're not quite able to localize where a sound source is coming from in a multi-talker scenario. And this can also lead to reduced auditory attention, again, because your lack of ability to separate the sound scene. So what are ways that we can personalize an HRTF for you or you? So the gold standard is to really just measure it. So we have in our lab this big arch, which has a bunch of different speakers in it. And then we have this turntable and chair. And then we can put 2 mics in the ear canal. And then from that, we can play a bunch of frequency sweeps from each of the speakers and then record how the sound is transformed at each of the microphones within the ear canal. It's pretty lengthy though, it takes about 20, 25 minutes. It gets even longer if the subject sort of moves around during the recording. Because of this, that's sort of susceptible to errors as well. Some people are really good at sitting still. Some people tend to like start slouching halfway through. We do have a tracker on their head to try and get them to, you know, get back into position, but it is very difficult and sometimes it's not doable. But we have captured a lot, and we can get, you know, quite a good resolution in terms of space around the subjects. But, you know, there is still issues with it. So another way that we could try and get someone's HRTF is to synthesize it. And we can do this by taking a 3D scan of their head and their shoulders and simulate it with a software called Mesh2 HRTF. But it is a bit computationally expensive. It does require us to pre-process the scans as well, which takes a bit of time. So where does my work come in? Well, firstly, I want to figure out, okay, how much do measured and simulated HRTFs differ numerically and also perceptually, because really perceptually is what counts. And then if they don't differ that much, can we replace measured HRTFs with these synthetic ones instead? So this is how I synthesize the HRTFs. So we take these scans from the Sonicom HRTF dataset, which is currently public, so you can grab these scans for yourself. Here is one of Lorenzo with the bold cap on, and you can see sort of it's got his polo on as well. And what we do is we behead him, and then we make him a bit more bold and get rid of sort of any facial hair to really just simulate, you know, where the skin is. That works in the Mesh2 HRTF software. And we also sort of plug the ear canal as well, because we want to really simulate at the entrance of the ear canal rather than like anywhere underneath. And it also kind of keeps things consistent between subjects, because not all subjects have like the exact same amount of torso being scanned, or also not the exact amount like within inside the ear canal, because you can't always really get in there. And from this, we can generate an HRTF that looks pretty HRTFE. So I've been working on, with the new sort of updated data set of this version, I've been working on synthesizing all of these HRTFs. So I've been making a lot of bald heads. So far, I've done like 124. And from that, we're synthesizing all the HRTFs from them as well. So in the current public data set, we've got 200. We're going to measure another 100 HRTFs and then hopefully synthesize a certain amount within that, and that will be released in the new data set, which will hopefully come at some point. So just a bit of a plug there. And hopefully you guys might find it useful. The 3D models themselves, because they've been pre-processed, can be easily manipulated if you did want to adjust them, resimulate them, and see how they might affect someone's perception. So are there any differences between the HRTFs numerically? So one thing that we can look at quite easily is the ITDs and the ILDs. And really hypothesized that there won't be that much difference, just because really it's the distance between the two ears that seem to impact these cues the most, and they should be represented pretty well by the 3D scanner of the head. And so here are two somewhat complicated graphs, but hopefully they're not too bad. So each dot is the difference between the synthetic and the measured at each location in elevation and azimuth. And sort of what it generally shows is you can sort of see these two little like horn-like structures in both the ILD and the ITD. And what it shows is that the synthetic HRTFs seem to underestimate, so they kind of create a smaller ITD and ILD difference compared to the measured. And I think this is likely due to the fact that with the 3D scans that we're using, we like behead them, so they don't incorporate the shoulder and the torso information that is like, well, is present in the measured conditions. So if we're seeing any errors, it'll probably be likely in these regions, but in front and above, like, it seems to be totally fine for the synthetic HRTFs. When we look at the log spectral distortion, so when we're trying to look at, the spectral content at each location and how that differs between the measured and the simulated, we can look at the left and the right ear, and they're pretty low. This is, again, across all of the HRTFs. They're sort of within like 3 dB. We do see lower log spectral distortion for the contralateral side, which kind of makes sense because the contralateral side is the side that gives the least information really to the HRTF. And we also see the most kind of in this below. Again, it's sort of in this bottom region of the HRTFs. And when we look across frequency, again, this is what others have found. It's the high frequencies that seem to be the ones that mismatch the most with the measured. And this could be likely due to the fact that with a 3D scan, you're only simulating the skin material. But when you're measuring someone, they've got hair, they've got clothes, they've got all of these absorbent materials that are likely to change the way that the high frequencies are simulated or measured. Okay, so what does this mean perceptually? So one way I've been testing this is localization. That's a pretty easy way to test how good an HRTF is. If you can render a sound source location realistically, someone should be able to localize where it comes from. And so I've been using a test sort of half developed by Rapalas in the lab where we present sounds both free field in this big dome and then also binaurally over headphones, and we present sort of visual cues using a MetaQuest, and that way we can present visual cues in all directions and also record their responses in all directions using the triggers, which before I've been in labs where we really struggled to like record in all directions. We kind of were limited at -90 and 90, whereas the VR kind of helps a lot with that. And the aim of this was to try and make like quite a quick test to be able to find any differences between things like generic and individual HRTFs, because some labs were doing like 8-hour experiments, which I really felt really sad for the subjects because they're like localizing broadband noise bursts over and over again for 8 hours. We didn't want to do that. We wanted it to be under an hour and we wanted it to be quick and sort of straightforward. So the one difference that I sort of did was training. So there is a level of procedural learning that has to happen. People don't usually localize noise bursts in an anechoic room, in silence, particularly when you get like the average Joe off the street. We might be quite good at it, but some other people aren't. And so how do we train them? Do we use their own HRTFs? Do we use a generic one? We didn't really want to push the learning into a specific HRTF, so we used the free field. So we presented free field sounds from the speakers, and then we got them to respond where they thought it was, and they got feedback as well in the form of a colored arrow. So if they were really far, they got a red arrow pointing the direction and where it should have been. And then they also got correction trials as well. So if they got too far away from the correct location, then they got 2 more goes afterwards. And once they did the training block, they then did 3 more blocks where in each trial they were given like a KEMA, a measured or KEMA, a measured or synthetic HRTF. These were like interleaved between each trials, and they had to again respond where they thought the noise bursts were coming from. And we only did 3 repeats. When I looked at repetition as an effect, they didn't really seem to get better as the test went on. When the test went on, so it seems that like a 45-minute experiment seemed to be totally okay, which is good. There's only 9 subjects so far. We unfortunately had to disassemble the big dome and then put in a new dome, which has only just gone in. So I'm going to get more subjects, hopefully around 20, but hence the sort of interim results that I'll be showing you now. So here is some just raw data. Here are the 4 different conditions. This isn't quite directly comparable, but I'm showing it anyway. So this the free field dome is called the Thunder Dome, sort of just for fun, but this is the free field condition, and then this is the KEMA, which is the generic one, and then this is the measured and this is synthetic. And this, in this plots, it's limited to an elevation of 0, so just the horizontal plane, just for ease of viewing, with the target on the bottom axis, so you can see it's like binned to 30 degrees, and the response on the Y. And you can see for the Thunderdome, people did pretty good, but when you get to generic, people make these sort of front-back confusions, which are less apparent in the measured and synthetic. The differences aren't so apparent here, but when you look at the elevation plane, so again, when azimuth is just 0 and you kind of look all the way around, you see the differences quite starkly. For the Thunderdome, we didn't have that many speakers directly in this plane, which is why there's only a few. But for the KEMA, you can see people responded pretty much everywhere, so not super great, but then less so for the measured. Again, the responses weren't as accurate as when they localized laterally, which again, as expected, the ITDs and ILDs are really salient cues, whereas these binaural spectral cues aren't that salient. But they still do better, and the same with the synthetic. But let's actually look at quantifying these. So this is the signed lateral accuracy and also the unsigned, and then the polar accuracy as well. And when we look at the signed lateral accuracy, you see no difference. Each dot, by the way, is a subject. And so we don't see like any bias or anything. When we look at the absolute errors, we see significant differences between the free field and then the binaural renderings by about like 5 degrees. People seem to have an error. And then when we look at the signed polar accuracy, we do see a bit of bias, but it's the unsigned polar accuracy where we see the biggest differences, where free field, they're able to do pretty well at under 10-degree errors, but with the generic, they really struggled with about 35-degree errors, but they were significantly better with the measured and the synthetic as well, suggesting there is a benefit for using these. Yep. Oh, sorry. So like the error, like if they were to the left of the like target response, I think it would be minus, and then if it's to the right was plus. So then that would be the sign, and then if it's unsigned, it would always be like 90, dependent on the direction. That's right. Yeah, it's just a way of like measuring bias or like just error in general. Cool. And so another thing that we look at is the confusion rate. So I can't remember exactly who classified what was a specific confusion, but it's kind of a nice way to be like, okay, if they made an error, what kind of error was it? Was it an error that was like pretty close to the target, or was it an error that was like a front back error? And so with this one, we looked at the precision error. So they had an error, but was within 45 degrees. So kind of a higher precision error is better. And we can see that with the training, the free field, they get, pretty good. They're around 95%. But with the KEMA measured and synthetic, with the KEMA are the worst, and then they're better with the measured and synthetic. What's probably most telling is the front-back confusions. With the free field, they barely make any front-back confusions. 3% of trials, they made an error, but with 20% of trials, they made these front-back confusions with the KEMA, and much less so with the measured and synthetic. And so this sort of generally shows that, we do better at localizing sound with our own HRTFs, and that we can also seem to be able to substitute them with synthetic HRTFs, as there isn't currently any marked difference between them, which is kind of promising if we do want to use synthetic instead of measured. However, people don't sit around in rooms listening to broadband noise in silence in anechoic rooms, so we should try and do something a bit more ecological and see if this still like holds. And so one of the things is spatial release from masking. Again, coming back to this like cocktail party problem where you're trying to separate someone's voice from a bunch of different voices. Multi-talker scenario. And it also involves things like informational and energetic masking. Again, like we'll want to use spatial cues or like all the other cues that within sort of spatial audio to be able to separate a target talker. And this can help aid speech intelligibility. And so the aim to test here is like, do different HRTFs change your sort of ability to do the spatial release from masking? You could imagine you might be worse with generic ones because you're not as able to localize the sound source compared to maybe your own. And so we pulled a test from, that's been done quite a few times. This specific version of it was pulled by the team at Malaga. And the idea is that you get presented a sentence, hopefully it will play. And you listen to Ready Baron and go to Red 6 now. And so you press Red 6 now, and then you go continue. But this would be really easy, so we don't really want to test that. We want to add more talkers on. So the next sound has 2 talkers, and you should listen out for the person that says Ready Baron and then hear what they have to say. It will be quite hard because you're not listening it to over headphones, but you can try. I think you should have heard White 3. But then we don't test 2 talkers, we test 3 talkers because we find, well, previous research has shown that 2 talkers isn't like hard enough for people to really, to pull out the effect of spatial release from masking. So this is the 3 talker one. So yeah, it's pretty difficult. It does get easier when you listen to over headphones, particularly when the target and the masker are spatially separated. So in the actual testing, we give people some trials to practice. They try with 1 talker, then 2 talkers, and 3 talkers. And then afterwards, we test them with 3 different blocks. Each block is about 11 minutes long. And each block, there'll be a sort of specific target talker location. This is sort of to help them out, give them a cue. The talker should be, below, in front, or above, and that will like help them try and separate it out a bit mentally. But so far, I've got 13 subjects. A bunch of students collected the data for me while I was on holiday, which was pretty great. So, and there are 10 repeats per condition. We looked at HRTF, again, the generic, measured the synthetic. Also, there's talker gender. There's like the male voices, the female voices. We wanted to see if there was a difference in being able to separate there. Also, the target location and then the masker location. And also bearing in mind when you see the next plots is that chance performance is 4%. To be able to just wildly answer is really low. So, and it's also a very hard task. So the percent you correct you get is quite low, but it's definitely above chance. So again, this is very preliminary work and I still need to do the full analysis since I just looked at it yesterday. But here you can see it colored by each subject and the distance from target in elevation. And I did the stats on this and this came out as significant so far, where we see despite this all being rendered binaurally, we do see a benefit of spatial release from masking. People's performance does increase in being able to detect, the right color and the right number when the masker and target are separated in elevation. And we had like different separations of like them being co-located at 0, maybe one was at 0, one was at -50, or 50 and -50. And we did see a benefit here. When we like split it out by HRTF, again, this didn't quite come out as significant as of yet, but I'm still need to collect more data. But it might be that we don't actually see much differences, but here you can see each subject with each HRTF with the generic in blue, the measured in orange and the synthetic in green. And we might expect to really see the largest differences when we see the greatest spatial separation, because that's when the spatial cues will give the most enhancement in doing this task. But there are also other things that we can look at rather than basic performance. We can look at, did they, instead of like hitting the correct answer, did they just hit like a completely different color and number that wasn't even said? Or did they hit a color and number that's said by a masker? And that sort of tells you whether it's like informational masking or maybe energetic masking. And so you can look at kind of the responses in a bit more detail to see, do these vary depending on the HRTF? Okay, and so something I wanted to touch on that I'm also trying to get at in this current postdocs is, well, and also I've seen in other people's research, is like, how do you test other features of spatial audio? And one big thing is like automatic processing. So our brain uses like the auditory system, is a pretty early warning system. Being able to know when a new sound has come from and where it's come from is quite useful if you're on the street and there's like a bus or a car behind you and you haven't actually attended to it. Whereas all of these previous tasks are asking someone to like attend specifically to the thing you want them to do. But what if you want to be able to test automatic processing? Also, what about more abstract features? Like, how do you ask someone how immersive something is, or how externalized is the sound? Particularly, again, if it's just like someone just off the street that doesn't even work in acoustics or audio. How do you get them to have the same concept of immersiveness as the next subject and as of you? And how do you like reliably test this? And so something I've been looking at is neural markers with my sort of neuroscience background. And so we use things like listening effort a lot. That's used a lot in non-spatial hearing. And so it can be brought into spatial hearing and also, again, other markers of things like attention. So I'll go a bit into the physiological measures that we've been using at the moment. So yeah, electrophysiology, it works with measuring the electrical signals from like the heart or the nervous system, but we work with the brain, so we use EEG. In my PhD, we got to put electrodes on neurons and select and record from single neurons directly. Can't quite do that in humans as easily. I don't think anyone really wants to volunteer to get electrodes put in their brains, so we use EEG caps instead. And we can put gel in each of these holes and put electrodes on, and it measures the surface potential across the scalp. And we can have like a bunch of these different channels. I think this is a 164. There's many channels, and so with the more channels, you can sort of localize the electrical activity across the scalp. And so, for example, you might get more activity in the back, which would be something visual, or in the front, which is more cognitive, or in the sides, which is more auditory. Here's a picture of me with a cap on, and what we get is these sort of signals from each of the channels. And here you can see just from the broadband signal, you get some oscillations, some artifacts, some other bits and pieces, but we can pull out a bunch of different features. We can pull out event-related potentials, which I'll go into. Same with this mismatch negativity response. Oscillatory power is a key thing. If you run some frequency analysis, and certain oscillations can be associated with certain cognitive processes like focus or attention or sleep. You can really see it when people close their eyes during an EEG experiment, and you really just immediately see these waves in the signal. Also things like entrainment as well, and coherence, like do all of the signals across the brain start to oscillate exactly at the same time once you've introduced a new stimulus. And also the topography of the activity. Do we see again, an increase in the signal in the front or in the back, and does this change between rendering methods? So one key thing that we always look out for is these event-related potentials, and they are, if you take those signals and you epoch it around a trial and you just average them off together, you'll usually get something that kind of looks like this. You need quite a lot of trials. EEG data is notoriously noisy, but you can get something that also has a lot of research behind it that allows you to kind of interpret it. So each of these ERPs will look slightly different based on the modality measured. So I think this is an auditory one, but you can get visual ones that look a bit different. And you'll get these positive and positive negative peaks, with earlier peaks being more stimulus related and later peaks being more cognitive related. Now, in the context of what I've been looking at is to take these ERPs and measure them in an audible paradigm. And so an audible paradigm aims to look at a neural response to an adapted stimulus, and then the difference between that and a neural response to like a new stimulus. So for example, if I play a pure tone at you like 1,000 hertz over and over and over and over again, and then randomly intersperse it with like a higher pitch, we can look at your brain activity for that standard 1,000 hertz tone, and then also for the higher pitch tone. And what we hope to see is that because you sort of played this thing over and over again, your brain will sort of lose that novel response, and then the novel response will come in for that high frequency. So here you can see an example where they played 1,000 and 1,004, and the difference in brain activity is pretty much the same. But as you start increasing the spacing between the low and the high tone, you start to see a bigger difference between in the neural signals, and this is called the sort of mismatch negativity response. And people have used it a lot for being able to test perceptual discrimination, right? So you can be able to test like given like almost the just noticeable difference, but the just noticeable difference of kind of any stimulus, and you can kind of abstract it out, right? So you can change all of the acoustic properties. Well, I say acoustic properties, but for example, you could change the frequency content of it, but you could keep the location the same. And you can like change different frequencies, but then keep the location the same, and then change the location randomly. And in that way, you're sort of testing the discrimination of this location. And so that's what I've been trying to look at. So again, this is very preliminary. This is one subject. One time I tried to see if I could get some nice recordings out of them. So I presented broadband noise burst in front of the listener, and then 20% of the time I presented noise burst from a different location. And this is sort of to test like their ability to, or the spatial discrimination between 30, 15, 5, and 0 degrees. And so what we would like to see is that the locations that are further apart from the standard would elicit a bigger response, and then locations closer would elicit a smaller response. And here you can see the evoked response from all of the channels. You kind of see this nice like increase in activity at 0. And then when you look at the difference between the standard and the deviant, so the standard is flat, but then the standard and the deviant for the when it's 30 degrees apart is quite big, and it gets smaller as you go to 15 and then to 5. And so the idea being here that if you can measure the magnitude of the MMN for different locations, how does that change when you use different rendering methods? Is it the same with measured, synthetic, generic HRTFs? If it is, if the spatial discrimination is the same, then why are we seeing differences in localization? Is it because then there's your inability to localize isn't because they don't sound different enough, it's because you don't have the associated cues. But yeah, so just to summarize all of this work briefly, I found numerical differences between measured and synthetic HRTFs, and they're pretty minimal, but the largest differences were sort of behind and below the subject and also high frequencies due to sort of hair and clothing. We also found that localization accuracy was better for personalized HRTFs, but no different really between the measured and the synthetic. And preliminary results show a spatial release from masking effect for all of the binaural rendering methods used. No difference so far between HRTFs, but again, we're collecting more data for all of these experiments. And yeah, I want to do some future work with actually using some EEG as well to assess, the spatial discrimination, again, with these different rendering methods. And so just to be less preliminary about it, I want to briefly talk about this project that has been using EEG properly, which is Anthea, and this has been a collaboration with UCL and Ericsson, and this is when I really started doing a lot of EEG recording, whereas before I didn't really do much. And this was really to get at the automatic processing of, you're in a spatialized environment and can you hear a new sound appear despite you not paying attention? And this is kind of important for people that really don't have these spatial cues like those with hearing aids, because how are they going to detect these new changes in a spatial environment? And that's why sort of Ericsson wanted to get involved, because they wanted to be able to create a test where they could look at someone's ability to detect a change in a spatial environment so that they could adjust hearing aid programs and things like that. So to briefly just go over some of the aims, so we had like a two-part tackling approach of it. So one was to do some behavioral tests to figure out, okay, does spatialization actually help you detect a change in a spatialized scene? How does the number of competing sound sources affect that? And also, can the location of a target affect target detection? For example, would you be better and quicker at detecting a new sound source coming from in front of you compared to like behind or left or right or above? And then we wanted to also do passive EEG measurements to see if we could actually use EEG to measure any change detection in the brain, and whether it would also be modulated by the location of the target as well. So just to briefly go over the methods. So we presented these chimeric sounds, which were broadband D, to be able to have all these binaural cues, and also ISDs and ILDs, because we presented sounds from all around in the speaker dome, from above and below and all around. And there was sort of this onset of sound sources. We varied it from sort of 4 sound sources up to 8. And 50% of the time, a new sound source would appear midway through. And during behavior, people were asked to press a button as soon as they heard this new sound source. It was kind of difficult. It got quite difficult, particularly when you got to a lot of sound sources, because everything just sort of sounds like a wall of sound, and you think you hear a new thing, but actually it was like the other things getting quieter and getting louder. So it wasn't an easy task. So we were quite worried when we did the passive EEG, would we even see anything, because they're not actually attending to the sound. But behaviorally, they actually didn't do too badly. Some people did pretty bad, but we found that spatialization was, performance was enhanced when we spatialized the sound. So here you can see performance and the number of sources, and each subject is a color. And we found performance was greater for at least the smaller number of sources. And they kind of started to equate as you increase the number of sound sources. And this is likely due to you not being able to separate the sound sources as easily, because there are so many, and so it becomes sort of more mono. And we also found that it was only in the spatialized condition that we found an effect of sources on performance, and that also that people with high frequency or higher high frequency thresholds had an impairment. So we did like a quick high frequency audiogram just before, just to see, if people with poor high frequency hearing would struggle because maybe they don't have access to those high frequency spatial cues. We did find that was the case for the spatialized audio. And we also, I didn't show it here, but we found differences in reaction time data for when the target was above and behind the listener. People were a bit slower when it came to more sources, but nothing in performance. And when we look at the EEG, so there's been a lot of work that's been done looking at change detection in sort of non-spatial hearing, and so we wanted to apply it in spatial hearing. And we did this when presenting 6 sources, and we found this sort of change response that happens. So when you look at the RMS power across all the channels, when there was a change, so this sort of new chimeric sound happening halfway through versus no, we see it within like 200 milliseconds, which is pretty rapid given that people's reaction times was like at around just under a second. So the brain was quite quickly able to detect when this new sound source happened. But we didn't see any differences in the topography between change or no change, so it seemed to be pretty prevalent across all channels. When we looked at target location, we also didn't see any differences between the front or behind or left or right. We might expect to see, I don't know, maybe a smaller change response for targets behind or above, but that doesn't seem to be the case. And yes, I think that's what I was going to say on that. Yes, so just overall, a quick summary of this work that is in prep. The paper is being written currently. We see a benefit in spatialization in detecting orchard targets. We observe impairment in performance for those with high frequency thresholds in the spatialized condition. And we can use EEG to detect target onset, and this can be done before the behavior. We can't, with this paradigm, at least with the EEG, distinguish between target locations, but we think this is likely due to the fact that we used so many sources. In the passive EEG, we used sort of 6 sources, whereas we see a lot of benefit of spatialization for around 4 sources. So perhaps we would see a difference in the EEG signal with target location if we used less sources. And what we're currently doing is we want to see if we see the same response when we use binaural audio. So if we present the same scene over headphones rather than free field, can we see the same effect? And then Ericsson are looking at applying this test in hearing impaired to see if they can better assess hearing aid performance as well. And yeah, I'd like to acknowledge everyone in the team. Like, this is when we played balls recently, so I got a nice photo. It was summer as well, but yeah, and then everyone else that has sponsored it. But yeah, any questions?
Do we have any questions here in the room at all? No? Body's bamboozled.
Sorry.
So, go on then. Yeah.
Excuse me. Wonder what happens with all this localization when they're wearing hearing aids. I know that's not what you're measuring, but as you were to, it's kind of interesting, because with an open fit, you'd have the spatial cues first of all going to the ear canal, and a few milliseconds later, you'd have whatever's picked up by a microphone, typically behind the ear, and therefore not in the location we're used to.
Yeah.
Any thoughts on that? Or is that just a left field question, which isn't very fair?
Yeah, well, my knowledge of hearing aids is not that great. And so, yeah, from what I know is that they really struggle having any of these spatial cues. I think, well, so the team is working on the BEARS project, which is trying to get people with bilateral cochlear implants to use distorted cues. So, if they do have these distorted spatial cues, you could train them to better localize with them if possible. But yeah, I don't think obviously you want the whole pinner, because that is what really gives them that advantage in being able to spatially separate. But yeah, I guess maybe the idea is that you could maybe give them, I don't know how you would do it, but present them these cues in the form of an HRTF, even though they're not getting it because the mic is behind the ear.
Anybody else? Oh, Becky, yeah.
So you're like synthesized HRTFs. Are they much easier to make those than to measure it?
Kind of. It depends.
Like, why? What's the advantage of having the synthesized HRTFs?
You don't need an anechoic room, and you don't need a big speaker rig, and you don't need mics, you don't need amps. You just need a seat and a 3D scanner. And
Yeah,
then
but you do. Yeah, but you still need that, don't you? So is that much easier?
I think, yeah, it'd be cheaper, and it also needs less space. At least in London, space is a premium. So if you can just do it in like a tiny little box room, I think that is works quite well. I think the key thing that takes a lot longer is the pre-processing of the scans. But I don't know how much the whole rig cost at our lab, but I think it is a lot less than a scanner. Yeah. It's also a lot quicker as well. It takes like 3 minutes to scan someone's head. Yeah.
I think many of us have been subjected to a lot of science sweeps. Any other questions in the room? I've got kind of it's maybe a very silly question, but it goes way back to the start. And you said about you don't measure people's ear canals, so when you're doing the scans, plug up people's ears. I was wondering, like, do we have any idea about how the ear canal might actually contribute to localization? I presume you plug them up because you would put earphones in, but that's going to disregard if there is actually an effect. Does that make sense?
So there doesn't seem to be any direction dependent effect, which is sort of what we're looking for. We're looking at, how it's affected by direction. It's what I've heard. But I think there are some people that are looking at doing like MRI scans to really get like the whole ear canal, modeling it that way. But from what we know, that's why we're sort of putting the microphone sort of at the opening of the ear canals, because the ear canal doesn't contribute that much directionally.
Interesting. Yeah, I mean, I'm not sure how you would incorporate it if you're presenting from headphones anyway. But
Yeah, exactly. And that too. Yeah.
Yeah. But I was just wondering if it might have been an effect on why HRTFs, even personalized ones, are not like perfect.
Yeah.
Any other questions?
Well, the resonance moves around, doesn't it?
Yeah. Well,
Yeah, I mean, assuming the ear canal resonance moves around, because presuming not everyone's ear canals are the same length. Not that
Right.
But,
The same direction. Like some people's are like, very far, all around.
Yeah.
But you can't have a test where you don't listen for your ear canals normally. No. So you can't kind of avoid them, can you?
If we were able to kind of prove an effect on localization of the ear canals, how could we possibly replicate that in earphones when you're going to have a repeated effect? Unless you're kind of bypassing the ear canal completely somehow, if that makes sense. Well, no, I don't think it would either. But like, if it is a problem, then it will remain a problem. Is my thought anyway. Yeah. So I don't think we have any questions online. So unless there's any further questions in the room. Nope. Okay. Well, in that case, we'll thank Kat again for coming up. For excellent talk. Thank you.
Cool. Thank you so much.
Thank you.
I stopped sharing and
Yeah.